Well, it's really a pleasure to be able to introduce Adam Miller for the Genetics Colloquium today. I have known Adam for quite a while, since our postdoc days in the Pacific Northwest. And, really Adam has done fundamental work throughout his career. So he did his PhD at the University of Oregon with Tori Herman, where he studied delta notch signaling in photoreceptor fate choice in C. elegans. And then after this, he moved to Seattle to work with Cecilia Moens at the Fred Hutch, during which he did a lot of fundamental work for the community, as well as started his research program on electrical synapses. So Adam, for those of you who don't know, uses zebrafish to study electrical synapses, and he initiated a forward genetic screen to look for novel modulators of electrical synapse formation. And during that work, obviously, it became really important to understand the causative mutations that were causing the phenotypes he was seeing. And so he developed a pipeline called RNA Mapper that now the community uses broadly to identify mutations that are caused by ENU or other such mutations in forward genetic screens. We've used this quite a lot. It's quite useful, taking the time it takes to understand the causative mutation from months or years down to days. In addition, he developed screening platforms for reverse genetic screens using CRISPR, and this was all in addition to the work that he started looking at electrical synapses. After he started at the U of O as an assistant professor, he's been incredibly productive and incredibly collaborative. In addition to his work that he'll tell us about today, he's also contributed to a number of studies using single-cell RNA-seq to both define the developmental zebrafish at a single-cell level, as well as a new work that you should look into on bioRxiv, looking at the neuronal populations of the octopus optic lobe, which is really, really cool work. But today he's going to tell us about his latest work on electrical synapses, the dark matter of the brain. With that, I'll ask you to mute, make sure your microphones are muted, and I'll let Adam take over.
Thanks, Katie. It's really great to be here. And I have to apologize to everyone. I'm sorry I'm here by Zoom, but all I can say is COVID is complicated still. And I'm glad I have the opportunity to come and present in this format. And it's really fun. I love Madison, and I'm really, really, really sad that I didn't get to come out and see you all in person. So maybe sometime soon I can do that. All right, so let me share my screen and we'll get going. Also, feel free to jump in at any time. I'd much rather have a conversation rather than just show you slides. So jump in, ask me questions. I'm happy to interrupt at any time. I don't really have a real agenda today as far as, I hope we get through the slides, but I don't really care. We can get as far as we get, and that'll be good enough. So today I'll tell you about electrical synapses, but not just electrical synapses. I'll tell you about chemical synapses too. And I called the electrical synapses the dark matter of the brain because when we look at a neural circuit like that on the screen right now, and we maybe start thinking about synapses, or I say the word synapses, you all think about neurotransmitters and receptors. But there are also these really important electrical synapses, and they're really actually hard to find in the brain for reasons I'll tell you as we go along. And we're missing them, and we don't know really how they're built. And so that's a big part of the program in my lab, is trying to understand how electrical synapses are built in the brain. But in big picture, my group is really interested in how you wire together developmental specificity in neural circuits. So how do you get a cool neuron like this one over here, that's orange, sending out this really cool projection pattern that's going down into the spinal cord and coming into contact with particular other neurons and, say, making a chemical synapse with neurotransmitter release and neurotransmitter receptors, contacting other neurons and not making a synapse, or contacting other partners and making an electrical synapse. Okay, so that sort of framework is really what my group tends to think about in big picture. How do you actually get the wiring to be right to serve behavior ultimately? And today we'll really focus on the molecular mechanisms of electrical synapse formation. But at the end, I'm going to hint at this cool new emerging space of coordination with chemical synapses. And we'll talk about that at the end as we get there. So before I get going, though, I have to acknowledge all the amazing people in my group. So I've been here with my own lab for just about 6.5 years now, and I've just been incredibly lucky to have such an amazing group of people to work with through this time. And so I can't thank them enough. And of course, everything I'm going to show you is all because of them. And so they will come up as I show you any particular project as we go along, but it's all their work that I'm going to be talking about today. So yeah, so how do we really think about building synapses in the brain? Well, often there's this classic comparison with like a computer chip and your brain. And the truth is that there's obviously very huge differences between any kind of computer chip that we have designed and your nervous system. For example, you have an estimated 100 billion neurons and something like 100 trillion synapses. And that estimate of synapses is more stars than the Milky Way, more stars than in the Milky Way galaxy. So we all have this astronomical complexity. And again, my group is trying to get after how the specificity of neural circuits is actually created. And this EM does a perfect job of talking about the specificity that I tend to think about. So I've gone in, this is EM from the mouse cortex. I've gone in and colored this particular neuron here in green, which is postsynaptic, and it makes these cool spines, which are a particular cell biological structure where a synapse is forming. And then in orange, I've colored in a couple of presynaptic axons. And so you can see the synapses kind of forming here. But also what I want to point out, again, is all this gray space that is around that green neuron where synapses are not forming. So there's really dense membrane contact in the brain and only selective regions where synapses are really being formed. And my lab and what we're interested in is really the molecular mechanisms that allow you to build those very selective spaces. Okay, if we zoom out a little bit from that view of the synapse and look at a tracing of about 100 micron cube in this from this EM reconstruction, this is what it looks like, right? There's lots of neurons. You can see the cell bodies, lots of wires going in there. It's starting to look a little bit like the computer chip over on the left. But again, we really need to think about that selectivity of building those very specific synapses in the right place at the right time. And so in that area, how do we think about electrical synapses? And so here's one of my favorite EM views of an electrical synapse. And electrical synapses are really neural gap junctions. Okay, and so this region right here was the first hallmark in EM where people started saying there's a really interesting structure formed between neurons. And it's this membrane, electron density right at the membrane, where these gap junction channels are really aligned. And this is what they look like on EM when you have a high enough quality EM. And there's been a lot of attention paid to the gap junctions and the channels, and we'll talk about them here in a second. But today, really what I'm going to try to tell you about is essentially beyond the gap junctions. So what is the other set of molecules that are required for building these gap junction channels, electrical synapses, in the right place at the right time in the brain? And how do you coordinate with chemical synapses? So those are the big three topics that we'll cover today. And we'll start by first orienting ourselves to what the neural gap junction channels really are. And so first off, where are they in your brain? And the answer is they're distributed throughout your brain. You're using them right now in your visual system to actually look at the Zoom talk, your auditory system to listen to me talk, your cortex to think about it, your motor system to breathe and move around. They're distributed throughout your brain. We don't know exactly where they are throughout all of these regions. Lots of electrophysiology has been done to show that they are broadly distributed, but that's part of the dark matter title of my seminar, is that they're really hard to capture. So a lot of the big EM connectome reconstruction projects that are going on, basically they don't have the resolution to see the gap junctions on the EM, or they use fixatives that basically destroy the gap junctions. They've been notoriously hard to track down in all sorts of different ways. But there's two beautiful EM reconstructions that have really shown us exactly where they are. And that is in C. elegans, the full EM reconstruction of C. elegans, and in a rabbit retina. And in those two examples where the quality was high enough, 20% of the connectome was electrical synapses, and every single neuronal class had an electrical connection. And so these are really broadly abundant. They make up a big part of the connectome, and they're broadly distributed. And ever increasing evidence says that human mutations in molecules that make electrical synapses are tied to autism, epilepsy, neurodegeneration, myopia, et cetera. And so we really think that these are important, but understudied, and we don't know how they're built. We do know that what's right here at this membrane are these gap junction channels. And in the vertebrates, these gap junction channels are made from connexin proteins. And so these are 4-pass transmembrane domain proteins with N and C termini on the intracellular side of the cell, extracellular loops out here. And 6 individual connexins come together as a hemichannel, and those hemichannels couple, making gap junction channels that will allow ionic and small molecule communication between the connected neurons. And your genome encodes 20 individual connexin genes. 5 of them have been shown to make electrical synapses, although the main one is connexin 36, and that's what we will focus on today. So the gap junction channels and these connexins are what are really important for making those channels. And this is really, this next picture is how you should think about the gap junction channels that I'm showing you throughout. And that is that there are many, many channels. So what this is on the right is a broken open gap junction plaque and atomic force microscopy that basically has shown you the placement of all those channels in these big plaques. And there's like a crystalline-like array of all of those channels, and then you can see the hexameric nature of one of the hemichannels from the 6 subunits of the connexin coming together. So I'm going to show you a lot of immunofluorescence of gap junctions in the brain coming up, and you should really be thinking about these fields of channels that are connecting the neurons together as we view those immunofluorescent pictures. So that's cool. Electrical synapses really are these gap junction channels. But again, I like to think about the stuff that is beyond the channels themselves. And so I call it beyond the gap. And this EM is really beautiful. The EMs like this don't come around very frequently, but they're in this particular image, there's this beautiful electron dense space that's in the cytoplasm adjacent to those gap junction channels. And we like to think of this space as a complex mix of proteins that's involved in the formation, function, and plasticity of electrical synapses, yet the molecules and mechanisms of how those proteins might be functioning was really unknown as I started up this project. And so we've called this connexin adjacent space the electrical synapse density. And we really think of it as all the proteins that are really required for forming these electrical synapses. But when I started this project, we didn't really know how to break it open, right? And so how can we open up the molecular world of the electrical synapse and try to understand what builds electrical synapses in the right place and at the right time? And so to do that, of course, as Katie told you, we use zebrafish, and we just love this system for its accessibility. And you know, you can see this beautiful brain here labeled with rainbow. You can see all those neurons. And in particular, we love the Meldner neural circuit shown here. And so we've got nice transgenics to look at these particular neurons. We know a lot about their function and about their cell biology. And we really try to span genetics to behavior in our understanding of electrical synapses and how they're built and how they function within the neural circuit. And we think we can do all of that in these cool Meldner cells. So where are Meldner and what are they doing? So here's a little cartoon of a 5-day-old zebrafish. 5-day-old zebrafish are like real animals, right? Zebrafish come from India. They have to swim around in the river in the Ganges. At 5 days old, they have to hunt. They have to escape. They have to do all those fishy sorts of things. And so this 5-day-old cartoon over here, there are the Meldner cells. You can still see them in green in the hindbrain. And then they send axons all the way down the spinal cord. Our imaging cut off here, but those go all the way down. And Meldner is driving this really cool escape response shown here. So there's a poor little fish there trying to get eaten by that big scary thing, right? And to escape, you need Meldner. And Meldner's whole job in life is to drive that turn. And it's been studied for a long time. We really know all the circuit, a lot of the circuitry, a lot of what's going on with Meldner and driving that turn. And I'm going to show you a stripped down version of especially the electrical synapses and the way we think of them here. And so here's those two Meldner cells. There's two in every animal. They get lots of sensory input. One of the main sources is from the ear, basically, and these auditory afferents that make synapses on the lateral dendrite. These synapses are actually mixed. They have an electrical component and a glutamatergic chemical component. So I've blown that up over here. Every individual contact is both electrical and chemical. And we'll come back to that later. But these auditory afferents are making these synapses onto Meldner. Meldner then sends that axon down the spinal cord. And at each segment in the animal, it finds this inner neuron called colo, and it makes an electrical synapse with colo. Meldner's making lots of other synapses as well. I'm just showing you a simplified version here. Meldner also, for instance, makes chemical synapses with motor neurons that drive the turning behavior. But the electrical synapses are what we will focus on. And they're extremely accessible and useful for us. And one thing you're going to need to know about them is that there's a presynaptic connexin, connexin 35, and a postsynaptic connexin, connexin 34. So the hemichannels that are making those connexins are actually different on the pre and postsynaptic side. And that's a theme that's going to come up again and again throughout the talk. So keep that in mind. I'm not going to show you the evidence for that. We've published that in the past. Here's the beautiful thing that you can see with Meldner. Here's a nice cell body over here. We got a cell body and a lateral dendrite. Each one of these individual white spots is an auditory afferent that's making a synapse onto Meldner. So there's one there, 2, 3, 4. And you can see connexin 34 and connexin 35 beautifully co-localized at all those spots. Okay, we can't tell the pre and postsynaptic nature of 35, 34 here in light microscopy. I'll show you how we get after that later. But suffice to say, those are both nicely and beautifully co-localized here and extremely stereotyped and accessible. Down in the spinal cord, here's Meldner axon going through. And here is the colo interneuron, which is also marked by this transgenic. And there you see connexin 34 and 35. Okay, so an amazingly beautiful, accessible system. And such stereotypy that when I first saw this, I was a postdoc and I was trying to figure out how to study electrical synapses. I saw this and I said, oh, I can do forward genetics on that and find mutants that would perturb the formation of these synapses. And so that's exactly what I did. I did a forward genetic screen using ENU as a random chemical mutagen. And I found mutations by looking at the spinal cord pattern of connexin staining at these Meldner colo synapses. And I found a class of mutation that I call disconnect. This was a bunch of different mutants, but basically they lost connexin at these stereotype positions. I found another class of mutation that I called amped, and those had ectopic electrical synapses forming along Meldner axons. So that was cool. I was really excited about those mutations, of course. But then while I was a postdoc, CRISPR just swept through the field and changed everything. And we decided that we really needed to use CRISPR, and we came up with ways, as Katie said, to use CRISPR in reverse genetic screens as well. And so to make a long story short, we came up with CRISPR mutagenesis ways where we can inject the components of CRISPR into zebrafish embryos, targeting specific genes, and then we can look for phenotypes in those injected animals. And so here's a few names of genes, and we've lost those electrical synapses. So the point here is that this system is incredibly accessible, and we've got classes of genes that are important for building the electrical synapse, and classes of genes that seem to be putting the brakes on building electrical synapses. So we think it's a really useful system to understand how these electrical synapses are actually being built. And I'll dive deeper into a story about ZO1 today to tell you how we think this molecule is really working and how it changes our perspective on electrical synapse formation. So this is really a beautiful collaboration between my lab here at Oregon and the Pareto lab at Albert Einstein in New York. And Jen and Abby and Audrey, who I think may be here in the audience today, are the ones that actually did this talk. And Audrey was a research assistant in my lab and now is with you in Madison. And I don't see her name, but I hope she's here because I'm going to present some of her work right now. And then in New York, Alberto Pareto's group is really good at doing electrophysiology. And so I'll show you some electrophysiology on the mutants in the system here in a moment as well. So we're going to go diving into scaffolds. And that's what ZO1 is. It's a cytoplasmic scaffold that is really beautiful for thinking about electrical synapses. And so here's some staining. Here's antibody staining of ZO1, which is this intracellular scaffold. And look at how it beautifully co-localizes with these connexins. It's right there at each one of these individual club ending contacts. It's right there, perfectly localized at the Malvern-Collo synapses as well. And when we knock it out, ZO1, of course, goes away, but both of the connexins go away as well. And so it's a simple result in a way, but actually this is the first molecule which is really required for localizing connexins at electrical synapses. And so we're starting to build a bigger idea about what electrical synapses are, just simply from this particular mutant here. And remember that this is an intracellular scaffold. So we think it's hanging out intracellularly next to the synapse here, and it's required for localizing these connexins. So we next wanted to ask, well, are the connexins actually required for localizing ZO? Is there a reciprocal requirement there? We, of course, have mutations in all the connexins. And so here is the connexin 35 mutant. That's pre-synaptic connexin 35. It's gone from the synapse. ZO still localizes to these club ending synapses. But notice that connexin 34, the post-synaptic connexin, does not. And we'll come back to that in a moment and think about it. But when we knock out the post-synaptic connexin, it's gone from the synapse, as it should be since we knocked it out. ZO is still localized. Connexin 34, or rather presynaptic connexin 35 is no longer robustly localized at the synapse. So the thing about that is interesting about this for me is that it really reveals that there's a hierarchy in building the electrical synapse. And that is that ZO1 is in charge of getting both of those connexins, the presynaptic and the postsynaptic connection to the synapse. They are not involved or not required to get ZO to the synapse. But then there's a mutual requirement for them to localize to the synapse. And remember that one is exclusively postsynaptic and the other is exclusively presynaptic. So there's actually cell-cell interactions required for building those gap junction channels just between those connexins. And then the ZO intracellular scaffold is required for getting those there and localizing them. Okay. So there's this hierarchy to building the electrical synapse. How is ZO actually doing it? And so how does ZO control connexin localization? ZO1 is part of a big class of molecules called MAGECs. These are scaffolding molecules. They're involved in all sorts of building intracellular connections in many different proteins. And so, for instance, these molecules up here are really important for chemical synapse formation in the postsynaptic density. And so ZO1 has lots of protein-protein interaction domains. We're not going to pay attention to most of them, but we will pay attention to these PDZ domains because essentially we hypothesize that ZO1 might directly interact with the electrical synapse connections. Okay, so if we look at one of these PDZ domains, I've just aligned the amino acids between your ZO1 and fish ZO1 here, and they're essentially identical. There's a few little changes, but I highlight this beta sheet and this alpha helix because there's lots of crystal structures of PDZ proteins, and this beta sheet and alpha helix make a ligand binding pocket. And those ligands that stick in there tend to be the C-terminal end of other proteins. So we basically thought that maybe PDZ from ZO1 was interacting with connexin at its C-terminus. And so there's your human connexin 36, and here's the fish connexins that are the homologs. And especially right at the C-terminal end, they are exactly the same. And this little orange ligand that's in this crystal structure is exactly 4 amino acids long. It's those 4 amino acids that are in there that are making interactions here. So we wondered whether or not there was a direct interaction between these guys. So we cloned out the proteins, expressed them in cell culture, and indeed there is. So ZO1 tagged with M Venus will co-IP with connexin 34, shown here when expressed in hex cells. ZO1 with connexin 35 will interact there. If we delete these last 4 amino acids, we completely lose that interaction. So it's a PDZ dependent interaction. And we've done lots of in vitro bacteria expressed proteins and everything else. It turns out that it's PDZ1 that's interacting with the connexin C terminal tails. So that interaction is there, but that's all in cell culture. What about in fish? Is there an interaction in the animals? And so that's shown here. We took zebrafish brains. We used ZO1 to do the IP. There's ZO1 coming out, and connexin 34 certainly comes out. In the ZO1 mutant, ZO1's gone, the connexin 34 is gone. But what I really want to draw your attention to is here in wild type, we don't see connexin 35 in wild type. Right, this is a background band down here. We should see connexin 35 right here. We see when we overexpress these proteins in vitro that ZO1 and connexin 35 can interact, but apparently not from the brain. So we wondered what was going on there. And what we thought was, hey, there's asymmetry. Connexin 35 is on the presynaptic side. Connexin 34 is on the postsynaptic side. Maybe ZO1 is on the postsynaptic side too. And so we wanted to test that idea. And so we went back to the fish to test this idea, and we combined 2 things to do it. So first off, we wanted to tag endogenous ZO1. So we used CRISPR engineering to put in a V5 tag at the endogenous locus of ZO1 in fish, so that we could know that we were seeing that particular ZO1. And we wanted to know where it was distributed in Maltner to see if it was postsynaptic, as we thought. We needed to combine that, though, actually with transplants, such that this cell is the only one making V5 tag ZO1. And so we do that through these transplants. So early embryos, we can basically use a transgenic that is tagged ZO1 and GFP positive, suck those cells up, stick them into a host, and let them develop. And we can get Maltner that came from the donor, while the rest of the circuit came from the host and didn't have ZO1 tagged or GFP tagged. Okay, so combining these 2 things, we could find the distribution of where ZO1 was within the Maltner cell. And I just want to remind you that Maltner is doing something cool here. I told you about the asymmetry at the synapse with connexin 34 postsynaptic. Well, Maltner is postsynaptic to these auditory afferents. So it's using connexin 34 there. But the auditory afferent is contributing connexin 35 presynaptically. So Maltner is sending connexin 34 over here. But down the axon, Maltner is presynaptic to these colos. And the colos are putting in connexin 34. So Maltner is actually sending 34 one way and 35 the other way. Okay, and we wanted to know what ZO1 was doing in that space. So here's the experiment finally. And that is that this green neuron, we're looking at the synapses over here, which is right over here in this space. And you can see that, hey, V5 staining shows us ZO1 beautifully localized to these club ending synapses. And remember that this ZO1 is coming from this green neuron, that is from Maltner. And if we follow this same Maltner down to the spinal cord, we know it's making V5 because it's putting it in its dendrite, but it's not putting it down in its axon. So V5 is exclusively postsynaptic here. And we've done all sorts of other transplants. We've made the auditory afferents V5, ZO1 B positive. We made the colos V5, ZO1 B positive. The answer is that ZO1 is always postsynaptic. So it's exclusively postsynaptic at these synapses. And we can actually ask whether or not it's functional postsynaptic. Of course, you expect it to be since it's localized there, but we can remove it specifically from the postsynaptic neurons by changing our transplants. So now the transplanted donor cells are ZO1B mutant. So the Maltner cell is lacking ZO1B, while the rest of the animal has ZO1. And so that result is shown up here. Just the control, we get the synapses. When we remove ZO1 from Maltner, the synapse connexins are no longer localized. And so ZO1B is localized postsynaptically, and functioning postsynaptically to build the electrical synapse. So the molecular asymmetry is at the level of the gap junction, but also at the level of the scaffold. And we think that the scaffold here is directly binding to that postsynaptic connexin to localize that postsynaptic connexin, which remember then is reaching across to the neighboring cell and localizing connexin 35. So there's an asymmetric building that actually is intercellular that is going on here, we think. So that's the idea of ZO1 for us in building these electrical synapses as we understand it at this point. What about synaptic function and behavior? So we seem to have lost connexin localization. Does it actually affect the synapse function and the behavioral output? And so Maudner is a beautiful system for doing electrophysiology. And this is done by Fabio in Alberto Pareto's lab. And you can stimulate the auditory afferents and then record from Maudner. And remember that these are mixed synapses, part electrical, part chemical, which is beautifully shown here. Here's the fast electrical component and the slower chemical component. So I didn't say it earlier, but in fish, electrical synapses are faster than chemical synapses. Okay, and you can see that separation really beautifully here. Okay, so what happens in the mutant? I tried to line these up perfectly for you. We should have a really big electrical component right here, and it's completely lost. It's a beautiful separation of function because the chemical synapse component remains. And we know this is glutamatergic chemical synapse component because you can put on these drugs that block glutamatergic transmission and get rid of that component as well. So we've really specifically lost the electrical component of transmission at these mixed club endings, which is really cool. What does that do to behavior, and what do we see? So if you just look at wild type startle responses over here, this is a montage of a fish that's turning its head and starting to swim away. And look at the timestamps here. This final one is 16 milliseconds. It's super fast, right? It's just lightning quick. You have to have a 1,000 frame per second camera to actually capture these escape responses. And in the ZO1b mutants, they start turning, but they're slower and they have coordination defects. And so these animals still have some of their chemical synapses intact, but they've lost their electrical synapse components, and they now have defects in the performance of their startle behavior. And I just want to remind you that that really matters, because if you're not fast and you're not coordinated, then you're lunch, right? And it's true of this Maudner circuit that you really need speed to be able to escape from the predator that's trying to eat you. But I think this is a general principle that at the end of the day, you need coordination of your electrical and chemical synapses to really get your final output of neural circuit computation. Okay? And these electrical synapse mutants are starting to show us some of how that's working.
Adam, could I interrupt with a question?
Of course.
It looked like on the last slide, the chemical synapse was strengthened in the knockouts. Is that systematic, and is it indicative that the electrical synapse is shunting the chemical synapse, or maybe some sort of adaptation?
It's a great question. We actually expected a lot more effect on the chemical synapse. And you're right that this particular recording, the chemical synapse does look elevated, that response. But the ensemble of many recordings, we saw no difference. And so we expected there to be a lot more effect on the chemical synapse, and we just didn't see it. So at least in this system, it really separated quite cleanly.
Oh, thank you.
Okay, so what did we learn here? What we learned is that we really think that ZO1 is the first molecule that's part of this electrical synapse density. It's localized here in the cytoplasm adjacent to the gap junction channels. And it is in charge of actually getting the channels there for structure and function. And without it, you don't build these electrical synapses. And I showed you for the Maudner circuit, that's true, but this happens throughout the brain. And we see defects in electrical synapses broadly. Asymmetric, ZO1 is asymmetrically required. and it's postsynaptically localized where it's functioning. And then there's this transsynaptic effect, i.e. between the cells, in which the connexins are working to localize the other connexin in the neighboring cell. And yeah, the ZO1 is required for function of electrical transmission behavior. And so what else is actually in this electrical synapse density? And that's a huge question for our lab. It's ongoing. We don't know all of the answers. One obvious thing is, hey, what about the presynaptic electrical synapse density here? And a new graduate student, William in lab, thinks that he has found the new scaffold on this side. And so that's emerging work that we're trying to figure out right now. It looks pretty good. But we imagine a much richer proteomic space. Cell adhesion molecules that are directing where these synapses are actually forming in the brain, regulators that control connexin localization and homeostasis and plasticity, all sorts of things that are going to regulate in complex ways how you build the structure and function of these synapses. And we're doing a lot of genetics and proteomics to actually unlock this electrical synapse density. And so that's work that's ongoing. You'll have to invite me out again in 5 years and I can tell you all about that. So let's move on to the next topic, which is subcellular specificity. And the notion here is that electrical synapses are formed by neurons at selective places within their compartments. And so shown over here, just thinking about a generic neuron, it could send electrical synapse proteins up to the dendrite, it could send them to the soma, or it could send it down to the axon. And I've drawn them all together on this one neuron, right? This one neuron is sending those electrical synapse proteins throughout it, but different neurons do make selective choices. Some neurons only send gap junction proteins to their dendrite and nowhere else. Some send them only to the axon and nowhere else. So there are choices being made as to where they are compartmentalizing the placement of these electrical synapse components, but no one understands how those choices are being made by neurons. And this really matters to neural circuits, namely that if it's up in the dendrites, you might connect dendrites to dendrites, or soma to soma, axon to axon, or an axon to a dendrite. And all of those things have real effect on the computations. Different electrical synapse configurations can be excitatory or inhibitory. They can synchronize neural networks or they can desynchronize them, and they can offer alternative paths for information flow through a network. And all of those things are really, at the end of the day, dependent on where the neurons have selected to put those electrical synapse components. And so computation ultimately depends on where those electrical synapses are built, but we really don't understand the molecular mechanisms of the compartmentalization of electrical synapse proteins. Okay, so Anne Martin, a postdoc in lab, and Jane Kissinger, a research assistant, have been working on this problem. And basically, we got into this from the forward genetic screen. So if we step back, one of the disconnect mutants that came out of this screen is called neurobeachin. And neurobeachin, the phenotype really is shown over here. You can see a reduction in connexin in both of the connexins and ZO1 localizing to the club ending synapses here. So neurobeachin is required for the robust localization of all of the proteins that I've been telling you about so far. Okay. And it was exciting for us to start thinking about neurobeachin because actually in mammals, people had already found neurobeachin in mammalian cell culture. So number one, it has a cool name, neurobeachin. But number two, it was localized to post-Golgi vesicles. It affects chemical synapse formation such that if you get rid of it, chemical synapses are not robustly made. Specifically, the trafficking and localization of neurotransmitter receptors are disrupted, and you aren't localizing glutamate receptors to the synaptic membrane. And human mutations are linked to autism and epilepsy. So we were excited to think about how neurobeachin was affecting electrical synapses. And I'm just going to give you a summary mainly of Anne's work and then highlight some of the new findings that are really unique and interesting. And so number one, neurobeacon localizes and functions postsynaptically. I think you can all imagine how we did those experiments now, but it's functioning postsynaptically and it binds to ZO1, but not the connexins. Okay, so neurobeacon is binding postsynaptically to ZO1, and that's how we think it's building the synapse. The localization is different than our other electrical synapse molecules. And so this green outline is the Maudner cell body. We've removed the GFP from the transgene. This is the cell body. This is Golgi stain. And neurobeacon, you can see, is dotted all through that Golgi staining. So this is similar to what has been reported in mammals. And you can see neurobeacon throughout the soma, presumably in sort of Golgi stuff here. But then if you go out to these club-ending synapses, now you can see ZO1 that I've been showing you throughout. But neurobeacon is interestingly localized kind of around the club-ending synapses. We don't fully understand what this localization means yet. I'll give you our model at the end here. But it's adjacent to sort of where the gap junction plaques really are, but right adjacent. It's just neighboring. And we really think this is kind of architecturally expanding the notion of the electrical synapse. And I'll give you a model for that at the end. But it's definitely present there at these electrical synapses. I've kind of cartooned it over here at the edge. It's interacting with ZO1 and it's required for getting these things here. And so it certainly builds into our idea of the hierarchy. Right now, neurobeacon is in charge of ZO1, which is then in turn in charge of the connexins. But what about this notion of compartmentalization and the specificity of delivery of electrical synapse proteins? And again, we're thinking about Maudner dendrite using connexin 34 and ZO1 specifically out here, while the axon is using connexin 35. And so that's where Anne turned to this, those transplants again. And the transplants have just been really illuminating here. This is just wild type. Again, this neuron is a wild type, but tagged with ZO1. And so you can see it dendritically here at the club ending synapses and not presynaptically. This is what I showed you before. But what happens when you get rid of neurobeacon? Well, you decrease the amount of ZO1 that's localized to these dendritic synapses, and it shows up in the axon presynaptically. So that's really cool, right? I mean, ZO1 is escaping the trap of the dendrite, flowing down the axon, and then localizing specifically presynaptically at these synapses, which it never does in wild type. Okay, so neurobeacon is restricting ZO1 to the dendrite and localizing it to those club-ending synapses. This also happens for the connexins. So here we've got a GFP tag connexin 34, Maudner club-ending synapses here, not presynaptically. This is a transplanted neuron. The neuron is just invisible here. It's not presynaptic, just like ZO1 in wild type over here. But in the neurobeacon mutant, connexin 34 is diminished, and now it shows up presynaptically. Goes down the axon and shows up presynaptically at the synapse. So neurobeacon, this is totally new. No one has ever seen a molecule that actually is important for subcellularly localizing electrical synapse proteins. And neurobeacon is restricting them to the dendrite. And so neurobeacon is somehow a gate in dendritic trafficking. We don't know the mechanism ultimately of how it's working here, but we're intrigued with the fact that it's localized over here at the synapse. And so one possibility, this is just a model that is really based on work from cell culture. No one really knows how the proteins are trafficked to an electrical synapse in vivo, but the notion from cell culture is that there are microtubules which are heading out to the lateral edge of a gap junction plaque. And those microtubules are stabilized by adhesion at that lateral edge. The channels then basically get put into the membrane right next to the gap junction plaque, where they're going to become part of that big gap junction plaque that I showed you before. And so perhaps that lateral localization of neurobeacon has a really important role for building in ZO1 and connexin at the synapse itself in some way that we don't understand. Alternatively, it could be that on those microtubules, the notion again from cell culture is that the hemichannels are being carried on vesicles by motors towards the gap junction plaques. And neurobeachin, as I showed you, is localized on Golgi membrane stuff, including vesicles. And so we think that neurobeachin could be on those vesicles and important for an interaction with ZO1 that restricts them to go out to the dendrite. So perhaps this is really about dendritic trafficking directly. And that if you lose that restriction, then maybe you can go down the axon. We don't know. We don't know which way it's working. We don't know how. But the cool thing about neurobeachin is that it really is the first molecule that has shown something about the cell biological specificity of how you get electrical synapses proteins specifically to the dendrite and not down to the axon. Okay, so neurobeachin expands the hierarchy of our thinking about electrical synapses. Neurobeachin is now in charge of the other electrical synapse proteins. It's a mechanism for subcellular localization, restricting those proteins to the dendrite and not letting them get down into the axon. Again, we don't understand the actual molecular mechanism here, but Anne's working hard on trying to actually uncover how that really works. And I'll just point out that neurobeachin also functions on chemical synapses. And so we're interested in exactly how that interaction works and how neurobeachin is functioning on both electrical and chemical synapses. And I'll say a little bit more about that in just one second. So just the summary here, right, is that we've further expanded the molecular complexity of electrical synapses. We've got ZO1, which we think is this electrical synapse density protein. Neurobeachin, which is involved in some way, we think, in the trafficking of these molecules to the synapse and trapping them in the appropriate subcellular compartment. And so the final topic I'm going to dive into here is this notion of electrical synapse coordination with chemical synapses. So what about those chemical synapses and what's going on there? And I already showed you evidence that in the brain, there's a coordination and function of electrical and chemical synapses. So I'm just showing you the electrophysiology from the club endings again, where we had the electrical and chemical synapse components. But you can see this all over the brain when you record in different places. But if you look at this beautiful EM of a mixed electrical or a mixed synapse, here's again that beautiful gap junction plaque. Here's the chemical synapse bits over here with synaptic vesicles lined up. Basically, people would draw a line in the sand in between those two things and say, hey, these things are molecularly separate. That's the general notion, is that there are separate molecules that would control electrical and chemical synapses, and that there's really, they're just two separate spaces. And I think there are bridging molecules, and that's what Anne's real postdoc project is. The one I told you about just a second ago was kind of her pandemic, she did it project, and this is her real project, where she's really thinking about this space of trying to bridge electrical and chemical synapses. And perhaps there are molecules that actually are interacting with both electrical and chemical synapses. And I showed you that the club ending is this really cool space where both of those are found, right? We have this mixed synapse at the club ending. And so here, here is Anne's picture of that. The electrical synapse density here is marked with ZO1, and you can see that. I showed that to you before. The postsynaptic density of the glutamatergic synapse here is shown by staining of SAP102, which is a scaffold of the postsynaptic glutamatergic synapse. And you can see it out here. It actually looks kind of like that neurobeachin staining I showed you before. There are these particular outposts kind of around the ZO1 staining around it. Nevertheless, we can see both the electrical and chemical synapses of each one of these synapses. This is one synapse here. This is another synapse here. And of course, the cool thing is that when you have a neurobeachin mutant, both of those are defective. Okay. So this last section, I'm just kind of bringing up this idea that, hey, neurobeachin might bridge between the electrical and chemical synapse. And in fact, Anne has gone on to show that neurobeachin binds ZO1, the electrical synapse scaffold. It also binds SAP102. the chemical synapse scaffold. So in fact, it is a molecule that binds at both electrical and chemical synapse components. So there is a biochemical bridge of neurobeachin between these two spaces. My beautiful orange bridge here that's bridging between the electrical and chemical synapse. Do we know how that's working in any sort of way? No, but we think it really highlights a new space of perhaps these two types of synapses, which everybody thinks they're separate, actually have more in common than we might think. And we think that there might be a big well of bridging molecules sort of in this Venn diagram overlap. And neurobeachin might just be one of them. And so Anne's been doing CRISPR screening, basically to try to find molecules that affect both electrical and chemical synapses. And this is just a short summary to say that we found, so far she's found cell adhesion molecules that affect both electrical and chemical synapse formation, synaptic homeostasis molecules, SYNGAP, that affect both, and some RNA binding molecules that actually, when she disrupts them, affect both electrical and chemical synapse formation. So we think, sort of like I was arguing for the electrical synapse, that we don't know much about this molecular space. We think this is a cool new space where there might be bridging molecules that actually control both electrical and chemical synapse formation. And so Anne's ongoing work is really to understand how these mutations she's created are functioning molecularly and cell biologically to build both electrical and chemical synapses. Okay, so we've covered three big topics. Beyond the gap, exploring the molecular biology of electrical synapses and thinking about those scaffolds that are really driving electrical synapse formation. We thought quickly about the subcellular specificity and neurobeachin and how it was compartmentalizing electrical synapse proteins. And I gave you just a tiny hint that maybe there's a shared molecular biology between electrical and chemical synapses, starting with neurobeachin. And we think there are molecules that are actually going to bridge building both of those types of synapses. So with that, I'd just like to come back and thank all the people in lab who are really, really incredibly amazing. And they're the ones that did all this work. And I'm happy to take any questions. And I made it on time, which I'm pretty impressed by. All right. Maybe I'll stop share. There we go. Uh-oh, where'd we go? Oh no. Is everybody still there? I'm sorry, I can't see you anymore. There we are. We're back. I'll take any questions.
I got one.
There's a question in the chat.
Oh yeah, I see it. Ultrafast exocytosis when you mess with the structure and function of electrical synapses. That's a great question. Boy, do we not know. I would love to know that. And we really, it would be fun to try to uncover that. But we don't have the techniques in our lab to do it. And so maybe you can tell me how we would want to do that. And I would love to figure it out. That sounds fun.
Got one.
I'm ready.
All right. I'm curious if you could speculate a bit on the utility of having the electrical and chemical synapses bridged. Is it that proximity matters, or that there's some important communication between them, or is it just as simple as ensuring that the electrical synapse is there as a fail-safe for the chemical synapse? Or, you know, are these? Do you have any intuition about it?
Well, I mean, as I showed you with the behavior that we look at, right, it's not just that the electrical synapse is just redundancy for the chemical synapse, right? And in fact, it really depends on the configuration, cell biologically and the gap junction channel, how they contribute to network function. So I don't think about them as just redundant in any sort of way. But yeah, I can't really come up with a true, like, you know, what's the molecular logic of the redundancy or this idea of bridging molecules. But all I can tell you is that for a long time, people have understood that electrical and chemical synapse function has been related for sure. And in fact, actually, like beautiful work, for instance, from Leach, where they block early developing electrical synapses in networks that they can actually track all the way to adulthood. When they block the electrical synapses there, they know that there are defects in chemical synapse formation and function ongoing further on. So there's been this long developmental crosstalk and sort of idea that these things are intertwined in development, but no one really understands how or exactly why. And we just think that there probably are molecules that are touching both in some sort of way, probably complex ways. For instance, neurobiчин very well might be trafficking, right? Trafficking to one type of synapse and then trafficking to the other type of synapse. But perhaps there are more structural molecules that bridge as well. We don't know yet.
Hi, Adam.
Hi.
Really beautiful work. I just was thinking about those mixed synapses and wondering if all of the sensory inputs onto the Mausner have them, like the lateral line or somatosensory. Is it just auditory or all?
It's a mixed bag. And so Chuck's old EMs actually kind of tell the tale. Chuck Kimmel did a bunch of really nice EMs back in the day. And for instance, lateral line are purely chemical. And but there are other sensory inputs that are actually mixed onto there. And then there are other inputs that are purely electrical. So it really is a mixed bag, and there's a lot of specificity depending on the particular presynaptic neuron in which it's touching. Each one's unique.
Hey, Adam.
Hey, Audrey.
How's it going? I think if I heard you correctly, you said you have a candidate for a presynaptic scaffold.
Yep.
I was just curious, because I know we looked at various other, proteins. Is this one functionally, do you think it's going to be functionally similar? And on the protein level, do they have similar domains? And what do you think's going on there?
I'll just give you your credit where credit is due. It's probably ZO2, and you are the one that actually found it. Took us a long time to actually get there. And so the latest thing that we've been doing, which we really like, is rather than CRISPR targeting to knock out a gene and see a phenotype, we now inject into embryos just V5 tags. And basically we CRISPR in V5 tags and look at the mosaics. And by doing that, we can see cell biologically beautiful V5 patterns within the Mausner circuit. And you can be tricky about it. And because of the Mausner circuit configuration, you can tell who's presynaptic and who's postsynaptic. And when we tag ZO2, it shows up presynaptically.
Interesting. Okay, cool. That's surprising given what we had seen before. So that's awesome.
Yep. It's all thanks to you. Thank you, Audrey.
Thank you.
I had a question about neurobiчин targeting of these pre and postsynaptic components. So it was really cool to see the electrical synapse components mistarget to the axon and neurobiчин loss of function. Do the receptors, the chemical synapse receptors, also mistarget?
We're looking, we don't know. We hope so. Yeah, it's been a slog to make the reagents to do it. So just think about that experiment, right? Like the experiment to do it, we had to have the mutant, and then we had to have the transgene from the transgenic, and then we had to have the engineered tagged protein. And so it's taken us a while to build up those reagents, and Anne is just about there now for the chemical synapse side of things. And she's going to start doing those transplants really soon. But it's a unique combination in this system that actually allows us to get after that. And she's close on the chemical synapse side. We're just not there yet. We assume so. We think they're going to slip down into the axon as well, but we'll see.
And one more question about neurobichin. Do you think there's any potential for a role in retrieval as
Sure.
opposed to trafficking?
Yep. Yeah, it could be. And we don't know. Yeah, it would be great to talk to you about thinking about ways to actually test that out, but we don't know at this point. Yeah, but why not? It could be.
Yeah, I'd love to talk more. Other questions for Adam? Please don't be shy.
All right, well, thanks everyone. I'm glad I got to come, even if it was virtually. It's really nice to see you all.
Thanks so much for making the time, Adam. It's great, as always.
Thanks, everyone. I'll see a couple of you tomorrow.
Great.
See ya.